Hello, everyone, and welcome back to our conversation on translation. So, I just want to remind everyone what translation is about before we move on to the actual steps of it. So, just as a reminder, currently in this module, we are talking about or focusing on gene expression. And remember, that is where we're starting with DNA, our instructions for how to make proteins, using that to make RNA, and the RNA is actually going to be what the cell uses to build the proteins. That's our end goal, are the proteins. And so the process of using DNA to make RNA was transcription. And then the process of using that RNA to build the proteins, what we're talking about now, is translation. Okay. So remember, we've got our messenger RNA, our transfer RNA, and the ribosome. Okay. And remember, part of the ribosome is the rRNA. So all of the different RNAs made during transcription are being put to use here. But for whatever reason, we don't call out our RNA, probably because it's not acting on its own. It's part of a functional put together ribosome, which is the rRNA plus the small subunit plus the large subunit. But I do want to mention real quick, y'all, all living things, so all cells, prokaryotic, eukaryotic, they all have ribosomes because they all need to make proteins. Proteins are essential for life. But the prokaryotic ribosome and the eukaryotic ribosome are not the exact same thing. They are slightly different. So, for instance, their overall size, the actual complexity and protein RNA composition varies slightly between the prokaryotic and the eukaryotic ribosomes. So the prokaryotic ribosomes are smaller, and we refer to them as the 70S ribosome, which you see here on this picture, while the eukaryotic ribosomes are slightly larger. Okay. And so I'm not going to expect you to remember all the ins and outs of it. I just want to make sure that I point that out because when we move on to Unit 2 and we're talking about antibiotics and what they target to help us control bacterial growth and hopefully limit and eliminate infections, these differences in the prokaryotic and eukaryotic ribosome are going to be important because we don't want to take medication that's going to impact our ribosomes, right? We only want to specifically target the prokaryotic ribosomes since the bacteria that are making us sick are prokaryotic. Okay, we will circle back to that a little bit later in the course. So, again, just a reminder, we read the mRNA in sets of 3 codons. I refer to the messenger RNA as a coded message. The first codon is called the start codon, the last one is called the stop codon. And remember, each codon is going to tell the cell specifically which amino acid is needed at any given time. So in this example, methionine is first. It's actually always first. The second amino acid we need is proline. So, in order for this particular protein to be made, we need proline and only proline to be second. We need valine and only valine to be third. Glutamine and only glutamine fourth, serine and only serine fifth, etc. Okay, so most proteins are at least 500 amino acids. Okay, so this is a very specific and detailed process. We need a lot of things to go right in order for a single protein to be made correctly. And there are thousands and thousands and thousands of proteins in the cell that need to be made correctly. So now let's get into the new content of this particular video. And that's going to be, how do these key players interact in order to get the correct amino to the correct amino acid to the correct site so that the correct protein can be built. And that's a lot of correctness that needs to happen. So one of the first things I want you to think about is the idea of interaction. Okay, we've talked about messenger RNA, transfer RNA, and an intact ribosome that are going to have to work together. So if we need proline and only proline to be second, how do we ensure that that happens? Remember that the tRNA is the gopher. It says, oh, we need proline. Okay, it goes out, it grabs proline, and it brings it to the ribosome where proline will be attached to the next amino acid. So, how this actually happens is there are multiple transfer RNAs in the cell, and they are specific for a certain amino acid. So, that's like I said, there are over 20 amino acids, and so we have a large number of different tRNAs. So, if you look at this tRNA, it actually has like a T structure here, but notice what's happening on each end. So up here is the amino acid attachment site. So what that means is there's going to be an amino acid, maybe I was going to do proline, but let's stick to the actual example on this slide. So this one is looking at codon 1, which is our start codon. So recall the start codon is always going to be AUG. You need to remember that. The start codon is always AUG. That will always code for the amino acid methionine or MET, which is what you'll see on your assessments. And so this tRNA can go out and grab methionine and bring it back to the ribosome where the protein's going to be built. Well, how does it do that? How does it know exactly where to drop off this methionine? Does it need to be first, third, 297th? How does it know? Well, if you look at the bottom of the tRNA molecule, notice there is a 3 codon region right here at the bottom. So remember, on the messenger RNA, I stressed in the last video that those 3 nucleotides are referred to as a codon. The mRNA is the coded message, right? Well, it turns out the tRNA also has 3 important nucleotides at the bottom, but we refer to it as the anticodon. Okay, so let's write this down so that we don't forget it. The messenger RNA, remember those are the directions, that has codons. Okay, we read the messenger RNA in sets of 3 called codons. But the tRNA, which is going to say, hey, you need methionine, I am the gopher. I'm going to go get it and bring it back, but I need to know where to deliver it. So it also has a set of 3 um, uh, nucleotides. There we go. And we refer to it as the anticodon. But notice, y'all, the word codon is still in there because a codon is a set of 3 nucleotides, right? Which this is. So do you remember what anti means? I mentioned this in one of my other videos. It means like opposite. So think of base pairing, okay? Because remember, y'all, tRNA and our RNA are nucleic acids. Okay, so remember our conversation about DNA and RNA previously, how they're going to be anti-parallel and complementary. Remember those terms? Well, here they're going to be complementary. So if you look at this, and I'm going to write it off to the side here just because it can be a lot to look at and listen to me. I'm looking here at our start codon. So remember, the start codon is always AUG. So I'm just going to write that up top to keep all of my text in one place. So look at what the anticodon is on this transfer RNA. It's complementary. When we're talking about RNA, remember that A always pairs with U always pairs with A, and G always pairs with C. So this is really important because the tRNA says, hey guys, I got the methionine. This is what you need. Wait, when do I drop it off? Where do I drop it off? It's only going to be able to deliver this methionine when it encounters the codon that is complementary to its own anticodon. So if you look at the image, let's look at the second codon here, CUG. Will this tRNA carrying MET, will it fit here? No, because the anticodon starts with a U, and U does not pair with C. So it cannot drop off MET at this second codon because the tRNA and the rRNA, I'm sorry, the tRNA and the mRNA are not complementary. Okay, so the mRNA has a codon, and it can only bind to the transfer RNA that has the correct anticodon. That's going to be what ensures that the correct amino acid is brought to the correct spot at the correct time. Okay, so that's looking at mRNA and tRNA interactions. Okay, let's now look at the picture on the right, which is bringing together our ribosome, but also the messenger RNA and the tRNA. So I'm going to get a highlighter because there's a lot to look at in this picture. So right here, I'm going to highlight it and then I'm going to erase it just because it's going to get busy really quickly. 5 prime to 3 prime. Look down here. That's the mRNA. Okay, and it's even labeled for you right here. Okay, so there's our messenger RNA. Actually, I'm just going to change my highlighter color. Let's see if I can do this without it getting overwhelming. Here is my intact ribosome. So you're going to see this small subunit here on the bottom. Notice that's what the mRNA is physically attached to, the small subunit. But it is connected here to the large subunit. Okay, remember that's the factory. That's where all of the protein is going to be built. So what that tells you is if the ribosome is the factory where the building of the protein is happening, then we need those directions to be present there as well. Hence, the mRNA is attached. But we're also going to need our little gophers, our tRNA that are out and about grabbing the amino acids we need. They need to be able to dock here as well. And so that's what you see happening. I'm going to change to green. You'll notice right here in this P site, you can't really see that green. Let's try a different shade. If I need to, I'll just erase. You can kind of see that. So notice here is the tRNA. It's got that T shape. And we have 2 of them connected in this particular image. I'm sorry, 2 of them are bound. One's in the P site, one is in the A site. Now, I am not going to be testing you on the A site versus P site versus E site, but I am going to refer to them when talking to make sure we're always looking at the same thing. So, what's going to be happening is the amino acid that's attached to our transfer RNA in the P site, eventually those amino acids are going to be attached to the one in the A site. Okay? Because again, the goal here is to build our protein. Proteins are made up of amino acids. So, as the tRNAs are coming in with their amino acid, we're going to be linking those amino acids together. Okay, so notice we have all of our key players here interacting. Okay, so let's now kind of step back and break translation down into 3 big steps. The first step is initiation, because we're going to initiate the process of translation. So essentially, what has to happen here is all of our key players need to get together, right? We can't start unless everybody's present. So what's actually going to happen is we're going to have our small ribosomal subunit, and it is going to bind to the messenger RNA. So I'm just going to draw it like this. Okay, we're going to have our 5' end and our 3' end. So there's our messenger RNA. It's attached to the small ribosomal subunit first. Then what's going to happen? So remember, our first codon. Okay, and please remember I'm artistically challenged too, so I'm doing my best here. So what's going to happen now is the first transfer RNA that's always going to be carrying methionine is going to come into play. So there's that T shape of my transfer RNA. It's going to be carrying methionine. Let's see if I can write that small. There we go. And it's going to come in and it's going to bind to our start codon. So just to remind you, this very first tRNA, we refer to it as the initiator tRNA because it's in helping to initiate this process. It's always going to have the anticodon UAC. UAC. Because remember, in order for the tRNA to bind and therefore deliver its amino acid to the right spot, it's going to have to bind to a complementary codon on the mRNA. So our start codon was AUG, so the anticodon that fits it is UAC. Okay, so I'm going to just write that here as well, UAC. So why it's important that this sequence of events happens is we're getting our ribosome, at least part of it initially, attached to the messenger RNA. Then we're bringing in our transfer RNA. So we're almost ready to go. We don't quite have an intact ribosome, but we're close. But the reason this tRNA has to bind first is because it's going to allow the large ribosome to come in and bind. Okay, now like I said, I'm artistically challenged, so my image should look like the pretty publisher image there, although it doesn't. But notice how in this pretty publisher image, you can see that there's our start codon AUG at the top. There's the transfer RNA, the initiator transfer RNA, with its anticodon UAC. It's also carrying our methionine, and it binds specifically to this P site. And that's important only because I'm trying to show you that that tRNA needs to be there so that large ribosomal subunit can lock into place correctly. Okay? So, y'all, this is initiation. We have all of our key players interacting together. The messenger RNA is attached to the ribosome, specifically at the small ribosomal subunit. The initiator tRNA came in to bind, and then the large ribosomal subunit followed suit. So that's initiation. Now comes elongation. Okay, and there's a lot of text on this slide. I'm not going to read it to you. We're going to talk through the images. So let's start at the top. So notice we have our mRNA. Again, it's this red ribbon right here. Let's go ahead and label it. Okay, we have our start codon at the top, AUG. We already had our initiator tRNA there, which was carrying the methionine. That's always the first amino acid. And so now we're ready to move on. And so in this example, leucine is second. And so that second codon was CUA. If you go back and look at the codon wheel, it would tell you leucine. So that means we needed our tRNA gopher to go out and grab a leucine. It needed to come in and bind so it can deliver the leucine. And so notice its anticodon is GAU, which is complementary to the mRNA's codon. So now check out what's going to happen. Notice that the methionine is going to be added to the leucine. Okay, so when that happens, so go to the middle image, notice now that the tRNA that just came in carrying leucine, now it's got both of those amino acids. That means our initiator tRNA is now empty. So it can now go and run and grab other methionines because odds are we're going to need a methionine again in our really big protein. So since the initiator tRNA is empty, it gets ejected from the ribosome, and this full tRNA is going to scooch over. Okay, so notice in the third picture, that's what happened. So notice here's our tRNA that's got our methionine and leucine attached to it. The third tRNA is coming in. The codon GCC told the cell, hey, we need alanine. So a tRNA carrying alanine says, oh, hey, I got you. It's able to bind here because its anticodon is complementary to that third codon. It's going to dock, and the same thing's going to happen. This chain is going to get attached over here so that the most recent tRNA will have all three amino acids. That will make this guy empty, so it will get ejected because, again, we're probably going to need another leucine somewhere along the way. And then this tRNA will then move over, and the next tRNA will be able to come in. Okay, so that's going to happen normally at least 500 times, sometimes more, because there are some really big proteins in the cell. And so this process, or this step of the process, is called elongation because we are building or elongating our chain of amino acids. Now, I do want to introduce another word to you real quick, and that is oops, that was not the highlighter. Sorry, y'all. Let me erase that real quick. So the term that I want to introduce is polypeptide chain. Okay. And polypeptide chain is a chain of amino acids. Okay. And you might be saying, Chelsea, I thought we were building a protein. And we are. So what we're doing right now is we are linking together all the amino acids. Okay. So we are creating a chain of amino acids. And I'm going to abbreviate amino acids as AA just for simplicity. And so this chain of amino acids is not going to be folded, just a linear chain of amino acids. We refer to this as a polypeptide or a polypeptide chain. And essentially, what this is, it means there's no 3D structure. We say that it is unfolded. We only refer to this chain of amino acids. It only becomes a protein when all the amino acids are able to interact and form their specific 3D shape. Only when there is a 3D shape, which then allows it to have its function, can we technically refer to it as a protein. Okay, so in this process, we're not looking at the amino acids folding. We're literally just looking at linking them together. So, if we wanted to be scientifically accurate, we say we're actually building our polypeptide. Once this process is done, all of those amino acids will interact with one another and fold and form a very specific 3D shape that allows that protein to then go out and do its job. Because remember, structure and function go hand in hand. Okay, so when we do more practice problems, I'm normally going to say, what is the amino acid sequence made? Or what is the polypeptide sequence? Okay, and that's just that we can be differentiating between polypeptide and protein. Okay, remember, protein folded has a specific 3D structure, it is functional. Polypeptide is not folded and not functional. All right. So, how does this process come to an end? Well, remember, we hit a stop codon at the very end. And I told you guys in the last video, there's no amino acid called stop. And so there are 3 different stop codons that exist, and the same thing happens no matter which one the particular mRNA has on it. And so what's going to happen is the ribosome's moving along. It's reading the messenger RNA. It's reading those directions, and it hits the end of that gene. It encounters a stop codon, and since there's no amino acid called stop, what actually happens is a termination factor. Is going to be involved in this, and it is actually going to kind of tell the whole process, hey, we're done. Um, and so that's going to signify, or that is going to tell the, y'all, my mind is crazy. Let's pause, pause. This is called a release factor. Okay, I'm sorry. Release factor. I was looking at the word termination when I was talking and I misspoke. I apologize. Let's write that down. A release factor is going to be what binds to the stop codon. And so it says, hey, we're done. It's time to release everything so that we can go and start building other proteins. Okay, so when this release factor is reached, or I'm sorry, when the release factor binds, notice what's happening here, y'all. The ribosome is going to release the mRNA and it's going to detach. Okay, notice these subunits are now separated from each other. Notice that the completed polypeptide chain is also being released. And then that means the mRNA is also going to be released because the cell is going to need more than just one copy of this protein, right? We have hundreds and hundreds of copies of a given protein. And so when this process is done, everything releases from one another. things disassemble, and now the cell can go and use these materials to build other proteins. Okay? And ultimately, mRNA does get degraded. Once the cell has what it needs, it will get degraded. Okay? All right, let's go ahead and end this video here, and I will make another video that just kind of summarizes the key points of central dogma, also known as gene expression. So as always, let me know if you have any questions, y'all.